Cerebral Cortex. Chapter 152. Introduction. Cerebral cortex is also called pallidum and it consists of two hemispheres. Surface area of cerebral cortex in human beings is 2.2 sqm. Both the cerebral hemispheres are separated by a deep vertical fissure, deep furrow or groove. The separation is complete anteriorly and posteriorly. But in middle portion, the fissure extends only up to corpus callosum. Corpus callosum is the broad band of commissural fibers, connecting the two hemispheres. Surface of the cerebral cortex is characterized by complicated pattern of sulci, singular equals sulcus, and gyri, singular equals gyrus. Sulcus is a slight depression or groove and gyrus is a raised ridge. Histology of cerebral cortex. Layers of cerebral cortex. Cerebral cortex consists of gray matter that surrounds the deeper white matter. It is formed by different types of nerve cells along with their processes and neuroglia. It is not uniform throughout. It is thickest, i.e. 4.5 cm at the precentral gyrus and thinnest at frontal and occipital poles. According to Economo, the cerebral cortex is formed by 6 layers of structures. Following are the layers from outside to inside. Molecular or plexiform layer. Molecular layer has few small fusiform cells. It also contains dendrites or axons from cells of deeper layers. External granular layer. External granular layer consists of large number of closely packed small cells, which are round, polygonal or triangular in shape. Dendrites of these cells pass into molecular layer. Axons end in the deeper layers. Some axons enter white substance of the hemisphere. Outer pyramidal layer. Outer pyramidal layer is formed by pyramidal cells, which are of 2 sizes. Medium-sized pyramidal cells are in the outer portion and larger pyramidal cells are in deeper portion. Internal granular layer. Like external granular layer, this layer also has closely packed smaller cells, which are stellate type. But, the nerve fibers are more in this layer than in external granular layer. This layer contains many horizontal fibers, which appear as a white strip known as outer strip. Ganglionic layer or internal pyramidal layer. Ganglionic layer or internal pyramidal layer consists of pyramidal cells of graded sizes. It is well developed in the precentral, motor, cortex. Pyramidal cells in this region are otherwise known as bed cells or giant cells. This layer also contains cells of Martinati. Martinati cells are peculiar in that their axons pass outward towards the surface of the cortex. Fusiform cell layer. Fusiform cell layer is in contact with white matter of cerebral hemisphere. It is composed of closely packed small spindle-shaped cells. Parts of cerebral cortex. Cerebral cortex is divided into 2 parts based on phylogeny, evolutionary development of a species neocortex. Allocortex. Neocortex. Neocortex is the phylogenetically new structure of cerebral cortex. It is also called isocortex or neopallium. This part forms the major portion of cerebral cortex. Part of the cerebral cortex that has all 6 layers of structures is called neocortex. Allocortex. Allocortex is the phylogenetically oldest structure of cerebral cortex. It has less than 6 layers of structures. It is divided into 2 divisions, namely, archicortex and paleocortex, which form the parts of limbic system, chapter 153. Lobes of cerebral cortex. In each hemisphere, there are 3 surfaces: lateral, medial, and inferior surfaces. Neocortex of each cerebral hemisphere consists of 4 lobes, Figs 152.1 to 152.3 frontal lobe, parietal lobe, occipital lobe, temporal lobe. Lobes of each hemisphere are demarcated by 4 main fissures and sulci, central sulcus or Rolandic fissure between frontal and parietal lobes. Parieto-occipital sulcus between parietal and occipital lobe. Sylvian fissure or lateral sulcus between parietal and temporal lobes. Calloso-marginal fissure between temporal lobe and limbic area. Cerebral dominance. Cerebral dominance is defined as the dominance of one cerebral hemisphere over the other in the control of cerebral functions. Both the cerebral hemispheres are not functionally equivalent. Some functional asymmetries are well known. Cerebral dominance and handedness. Cerebral dominance is related to handedness, i.e. preference of the individual to use right or left hand. More than 90% of people are right-handed. In these individuals, the left hemisphere is dominant and it controls the analytical process and language-related functions such as speech, reading, and writing. Hence, left hemisphere of these persons is called dominant or categorical hemisphere. Right hemisphere is called representational hemisphere since it is associated with artistic and visuospatial functions like judging the distance, determining the direction, recognizing the tones, etc. Lesion in dominant hemisphere leads to language disorders. Lesion in representational hemisphere causes only mild effects like asteriognosis. Left hemisphere is the dominant hemisphere in about 75% of the right-handed persons. In the remaining left-handed persons, right hemisphere controls the language function. Some of these persons do not have dominant hemisphere. Brodmann areas. Brodmann area is a region of cerebral cortex defined on the basis of its cytoarchitecture. Cytoarchitecture means organization of cells. Brodmann areas were originally defined and numbered in 1909 by Corbeny and Brodmann depending upon the laminar organization of neurons in the cortex. Some of these areas were given specific names based on their functions. During the period of a century, Brodmann areas had been extensively discussed and renamed. Frontal lobe of cerebral cortex. Frontal lobe forms 1/3 of the cortical surface. It extends from frontal pole to the central sulcus and limited below by the lateral sulcus. Frontal lobe of cerebral cortex is divided into two parts: precentral cortex, which is situated posteriorly; prefrontal cortex, which is situated anteriorly. Precentral cortex. Precentral cortex forms the posterior part of frontal lobe. It includes the lip of central sulcus, whole of precentral gyrus and posterior portions of superior, middle, and inferior frontal gyri. It also extends to the medial surface. This part of cerebral cortex is also called excitomotor cortex or area, since the stimulation of different points in this area causes activity of discrete skeletal muscle. Precentral cortex is further divided into three functional areas: Fig 152.3 primary motor area, premotor area, supplementary motor area, primary motor area. Primary motor area extends throughout the precentral gyrus and the adjoining lip of central sulcus. Areas 4 and 4s are present here. Structure of primary motor area. Though this area has all the 6 layers, the granular layer is thin. Special structural feature of this layer is the presence of giant pyramidal cells called bed cells in ganglionic layer. Connections of primary motor area. Efferent connections. Fibers of pyramidal tracts arise from the bed cells. These fibers synapse with motor neurons in anterior gray horn of opposite side. Few fibers reach the same side motor neurons, in spinal cord. Frontopontine fibers from this area reach pontine nuclei of same side. Fibers are also projected to corpus striatum, red nucleus, thalamus, subthalamus and reticular formation. Association fibers connect the primary motor area to other areas of cortex. Afferent connections. Primary motor area receives fibers from dentate nucleus, cerebellum, via red nucleus and thalamus. Functions of primary motor area. Primary motor area is concerned with initiation of voluntary movements and speech. Area 4. It is a tapering strip of area situated in precentral gyrus of frontal lobe. Broad end lies superiorly at the upper border of hemisphere and most of the efferent fibers of primary motor area arise from this area, Figs 152.4 and 152.5. Function of Area 4. Area 4 is the center for movement American Samoa. It sends all efferent, corticospinal, fibers of primary motor area. Through the fibers of corticospinal tracts, area 4 activates the lower motor neurons in the spinal cord. It activates both alpha motor neurons and gamma motor neurons simultaneously by the process called coactivation, chapter 157. Activation of alpha motor neurons causes contraction of extrafusal fibers of the muscles. Activation of gamma motor neurons causes contraction of intrafusal fibers, leading to increase in muscle tone. Effect of stimulation of area 4. Electrical stimulation of area 4 causes discrete isolated movements in the opposite side of the body. The groups of muscles or single isolated muscle may be activated depending upon the area stimulated. Localization, homunculus. Muscles of various parts of the body are represented in area 4 in an inverted way from medial to lateral surface. Lower parts of body are represented in medial surface and upper parts of the body are represented in the lateral surface. Order of representation from medial to lateral surface: toes, ankle, knee, hip, trunk, shoulder, arm, elbow, wrist, hand, fingers, and face. However, parts of the face are not represented in inverted manner, Fig 152.6. Area 4 is concerned with contraction of discrete muscles. It sends motor signals to the facial muscles of both sides, bilateral, and the other muscles of the opposite side, contralateral. Effect of lesion of area 4. Effect of lesion or ablation of area 4 differs in different species. In cats, the ability to walk is not affected. In monkeys, there is contralateral flaccid paralysis, hypotonia, and loss of reflexes. Myotatic reflexes reappear in a short time. Recovery occurs only in proximal parts of limbs, but the digits remain permanently paralyzed. In man, the symptoms are severe than in monkeys. In unilateral lesion, paralysis occurs in contralateral side. Complete paralysis is rare. If both sides are affected, the effect is more severe. Recovery occurs very slowly. During recovery, upper parts of body recover first. If area 4 is affected along with area 6, the effect is very severe, causing hemiplegia with spastic paralysis. Hemiplegia means the paralysis in one half of the body. In spastic paralysis, the muscles undergo spastic contraction due to increased muscle tone. Area 4s. Area 4s is called suppressor area. It forms a narrow strip anterior to area 4. It scrutinizes and suppresses the extra impulses produced by area 4 and inhibits exaggeration of movements. Premotor area. Premotor area includes areas 6, 8, 44, and 45. The premotor area is anterior to primary motor area in the precentral cortex. The premotor area is concerned with control of postural movements by sending motor signals to axial muscles, muscles near the midline of the body. Structure of premotor area. Premotor area is similar to primary motor area in structure except for the absence of giant pyramidal cells in ganglionic layer. Area 6. Area 6 is in the posterior portions of superior, middle, and inferior frontal gyri. It is subdivided into 6A and 6B. It gives origin to some of the pyramidal tract fibers. The other connections are similar to those of area 4. Functions of area 6. Area 6 has two functions. It is concerned with coordination of movements initiated by area 4. It helps to make the skilled movements more accurate and smooth. It is believed to be the cortical center for extrapyramidal system. Effect of stimulation of area 6. Electrical stimulation of area 6 in human being causes the same effects as the stimulation of area 4. However, the stimulus must be stronger to evoke response from area 6. The effects of stimulation of this area are: stimulation of area 6 causes generalized pattern of movements like rotation of head, eyes, and trunk towards the opposite side. Stimulation of 6B produces rhythmic, complex coordinated movements involving the muscles of face, buccal cavity, larynx, and pharynx. Effect of lesion of area 6. Lesion or removal of area 6 in monkeys leads to loss of skilled movements. After the lesion, the recovery may occur, but the movements become awkward. It also produces grasping reflexes. Lesion involving area 6 and area 4 produces severe symptoms of hemiplegia with spastic paralysis. Area 8. Area 8 is called frontal eye field. It lies anterior to area 6 in the precentral cortex. It is concerned with movements of eyeball. This area receives afferent fibers from dorsomedial nucleus of thalamus and occipital lobe. It sends efferent fibers to oculomotor nuclei in tegmentum of midbrain. Function of area 8. Frontal eye field is concerned with conjugate movement of eyeballs, chapter 165. This area initiates voluntary scanning movements of eyeballs and it is independent of visual stimuli. It is also responsible for opening and closing of eyelids, pupillary dilatation, and lacrimation. Effect of stimulation of area 8. Stimulation of this area causes conjugate movements of eyeballs to the opposite side. Effect of lesion of area 8. Lesion of this area turns the eyes to the affected side. Conjugate movements of eyes are lost. However, pupils and eyelids are not affected. In animals, while walking, circular movements occur towards the affected side. Broca area. Broca area is the motor area for speech. It includes areas 44 and 45. Broca area is present in left hemisphere, dominant hemisphere, of right-handed persons and in the right hemisphere of left-handed persons. It is a special region of premotor cortex situated in inferior frontal gyrus. Area 44 is situated in pars triangularis and 45 in pars opercularis of this gyrus. Function of Broca area. Broca area is responsible for movements of tongue, lips, and larynx, which are involved in speech. Effect of lesion of Broca area. Lesion in Broca area leads to aphasia, chapter 162. Supplementary motor area. Supplementary motor area is situated in medial surface of frontal lobe rostral to primary motor area. Various motor movements are elicited by electrical stimulation of this area like raising the contralateral arm, turning the head and eye and movements of synergistic muscles of trunk and legs. Function of supplementary motor area. Exact function of this area is not understood clearly. It is suggested that it is concerned with coordinated skilled movements. Effect of lesion of supplementary motor area. During lesion in this area of human being, the head and eyeballs turn towards the affected side. Destruction of this area in monkeys causes weak grasping reflexes in contralateral side and bilateral hypertonia of shoulder muscles. But, paralysis is not noticed. Prefrontal cortex or orbitofrontal cortex. Prefrontal cortex is the anterior part of frontal lobe of cerebral cortex, in front of areas 8 and 44. It occupies the medial, lateral, and inferior surfaces and includes orbital gyri, medial frontal gyrus and the anterior portions of superior, middle, and inferior frontal gyri. Areas present in prefrontal cortex are 9, 10, 11, 12, 13, 14, 23, 24, 29, and 32. Areas 12, 13, 14, 23, 24, 29, and 32 are in medial surface, table 152.1. Areas 9, 10, and 11 are in lateral surface. Connections of prefrontal cortex. Afferent fibers. Afferent fibers of prefrontal cortex come from dorsomedial nucleus of thalamus, hypothalamus, corpus striatum, amygdala, midbrain. Areas 23, 24, 29, and 32 receive fibers from anterior nucleus of thalamus. Area 32 receives fibers from subpressor area of precentral cortex also. Efferent fibers. Efferent fibers are projected to thalamus, hypothalamus, tegmentum, caudate nucleus, pons, temporal lobe of cerebral cortex. Area 13, along with hippocampus, uncus, and amygdala sends fibers to mammillary body of hypothalamus via fornix. This area is concerned with emotional reactions. Functions of prefrontal cortex. Earlier, this area was considered as inexcitable to electrical stimulation. Hence, it was called the silent area or association area. but, now it is known that the stimulation of this area with low-voltage electrical stimulus causes changes in the activity of digestive, cardiovascular, respiratory, and excretory systems and other autonomic functions. It also causes fear. Various functions of prefrontal cortex are, it forms the center for the higher functions like emotion, learning, memory and social behavior. Short-term memories are registered here. It is the center for planned actions. This area is the seat of intelligence, so, it is also called the organ of mind. It is responsible for the personality of the individuals. Prefrontal cortex is responsible for the various autonomic changes during emotional conditions, because of its connections with hypothalamus and brainstem. Effect of lesion of prefrontal cortex. Bilateral lesion or removal of prefrontal cortex in human beings does not cause paralysis. It causes lack of initiation and loss of mental alertness. Very little or no change occurs in memory, judgment, and intelligence. Applied physiology, frontal lobe syndrome. Injury or ablation of prefrontal cortex leads to a condition called frontal lobe syndrome. Features of this syndrome are, emotional instability, there is lack of restraint, leading to hostility, aggressiveness, and restlessness. Lack of concentration and lack of fixing attention. There is lack of initiation and difficulty in planning any course of action. Impairment of recent memory occurs. However, the memory of remote events is not lost. Loss of moral and social sense is common and there is loss of love for family and friends. There is failure to realize the seriousness of the condition. The subject has the sense of well-being and also has flight of ideas. Apart from mental defects, there are some functional abnormalities also, hyperphagia, increased food intake. Loss of control over sphincter of the urinary bladder or rectum. Disturbances in orientation. Slight tremor. Parietal lobe. Parietal lobe extends from central sulcus and merges with occipital lobe behind and temporal lobe below. This lobe is separated from occipital lobe by parieto-occipital sulcus and from temporal lobe by sylvian sulcus. Parietal lobe is divided into three functional areas, sumaesthetic area I, sumaesthetic area II, sumaesthetic association area. In addition to these three areas, a part of sensory motor area is also situated in parietal lobe, see below. Sumaesthetic area I. Sumaesthetic area I is also called somatosensory area I or primary sumaesthetic or primary sensory area. It is present in the posterior lip of central sulcus, in the postcentral gyrus and in the paracentral lobule. Areas of sumaesthetic area I. Sumaesthetic area I has three areas, which are called areas 3, 1, and 2. Anterior part of this forms area 3 and posterior part forms areas 1 and 2. Connections of sumaesthetic area I. Sumaesthetic area I receives sensory fibers from thalamus via parietal part of thalamic radiation. Localization, homunculus. Different sensory areas of the body are represented in postcentral gyrus, primary sensory area, in an inverted manner as in the motor area. Toes are represented in lowest part of medial surface, legs at the upper border of hemispheres, then from above downwards knee, thigh, hip, trunk, upper limb, neck, and face. Representation of face is not inverted. Representation of parts of face from above downwards is eyelids, nose, cheek, upper lip and lower lip, Fig 152.7. Functions of sumaesthetic area I. Sumaesthetic area I is responsible for perception and integration of cutaneous and kinesthetic sensations. It receives sensory impulses from cutaneous receptors, touch, pressure, pain, temperature, and proprioceptors of opposite side through thalamic radiation. Area 1 is concerned with sensory perception. Areas 3 and 2 are involved in the integration of these sensations. This area sends sensory feedback to the premotor area. This area is also concerned with the movements of head and eyeballs. Discriminative functions, in addition to perception of cutaneous and kinesthetic sensation, this area is also responsible for recognizing the discriminative features of sensations. Discriminative functions are, spatial recognition, tactile localization, two-point discrimination and recognition of position and passive movements of limbs. Recognition of intensity of different stimuli. Recognition of similarities and differences between the stimuli. Effect of stimulation of some aesthetic area. Electrical stimulation of some aesthetic area produces vague sensations like numbness and tingling. Effects of lesion of some aesthetic area. If lesion occurs only in the sensory area without involvement of thalamus, the sensations are still perceived. But, the discriminative functions are lost. If thalamus also is affected by lesion, there is loss of sensations in the opposite side of the body. Some aesthetic area 2. Some aesthetic area 2 is situated in post-central gyrus below the area of face of some aesthetic area I. A part of this is buried in sylvian sulcus. It is also known as secondary some aesthetic area or somatosensory area 2. Functions of some aesthetic area 2. Some aesthetic area 2 receives sensory impulses from some aesthetic area I and from thalamus directly. Though the exact role of this area is not clear, it is concerned with perception of sensation. Thus, the sensory parts of body have two representations, in some aesthetic area I and area 2. Some aesthetic association area. Some aesthetic association area is situated posterior to post-central gyrus, above the auditory cortex and in front of visual cortex. It has two areas, 5 and 7. Functions of some aesthetic association area. Some aesthetic association area is concerned with synthesis of various sensations perceived by some aesthetic area I. Thus, the some aesthetic association area forms the center for combined sensations like stereognosis. Lesion of this area causes astereognosis. Sensory motor area. Sensory area of cortex is not limited to post-central gyrus in parietal lobe. It extends anteriorly into motor area in precentral gyrus of frontal lobe. Similarly, the motor area is extended from precentral gyrus posteriorly into post-central gyrus. Thus, the precentral and post-central gyri are knit together by association neurons and are functionally interrelated. So, this area is called sensory motor area. Function of sensory motor area is to store the timing and programming of various sequential movements of complicated skilled movements, which are planned by neocerebellum, Table 152.2. Applied physiology. Lesion or ablation of parietal lobe, sensory cortex, results in the following disturbances: contralateral disturbance of cutaneous sensations. Disturbances in kinesthetic sensations. Loss of tactile localization and discrimination. Temporal lobe. Temporal lobe of cerebral cortex includes three functional areas, Table 152.3. Primary auditory area. Secondary auditory area or auditopsychic area for equilibrium. Primary auditory area. Primary auditory area includes, Area 41. Area 42. Wernicke area. Areas 41 and 42 are situated in anterior transverse gyrus and lateral surface of superior temporal gyrus. Wernicke area is in upper part of superior temporal gyrus posterior to areas 41 and 42. Connections of primary auditory area. Afferent connections. Primary auditory are receives afferent fibers from, medial geniculate body via auditory radiation pulvinar of thalamus. Efferent connections. This area sends efferent fibers to, medial geniculate body pulvinar. Functions of primary auditory area. Primary auditory area is concerned with perception of auditory impulses, analysis of pitch and determination of intensity and source of sound. Areas 41 and 42 are concerned only with the perception of auditory sensation, sound. Wernicke area is responsible for the interpretation of auditory sensation. It carries out this function with the help of secondary auditory area, Area 22. Wernicke area is also responsible for understanding the auditory information about any word and sending the information to Broca area, Chapter 162. Secondary auditory area. Secondary auditory area occupies the superior temporal gyrus. It is also called or auditopsychic area or auditory association area. It includes area 22. This area is concerned with interpretation of auditory sensation along with Wernicke area. It is also concerned with storage of memories of spoken words, chapter 162. Area for equilibrium. Area for equilibrium is in the posterior part of superior temporal gyrus. It is concerned with the maintenance of equilibrium of the body. Stimulation of this area causes dizziness, swaying, falling and feeling of rotation. Applied physiology, temporal lobe syndrome. Temporal lobe syndrome is otherwise known as Kluver-Bucy syndrome. It is observed in animals, particularly monkeys, after the bilateral ablation of temporal lobe along with amygdala and uncus. It occurs in human beings during bilateral lesions of these structures. Manifestations of this syndrome are, aphasia, disturbance in speech, chapter 162. Auditory disturbances such as frequent attacks of tinnitus, auditory hallucinations with sounds like buzzing, ringing or humming. Tinnitus means noise in the ear. Hallucination means feeling of a particular type of sensation without any stimulus. Disturbances in smell and taste sensations. Dreamy states, the patients are not aware of their own activities and have the feeling of unreality. Visual hallucinations associated with hemianopia. Occipital lobe. Occipital lobe is called the visual cortex. Areas and connections of occipital lobe is given in table 152.4. Areas of visual cortex. Occipital lobe consists of three functional areas: primary visual area, area 17. Secondary visual area or visuopsychic area, area 18. Occipital eye field, area 19. Connections of occipital lobe. Occipital lobe receives afferent fibers from lateral geniculate body. It sends efferent fibers to superior colliculus and lateral geniculate body. Functions of occipital lobe. Primary visual area, area 17, is concerned with perception of visual sensation. Secondary visual area, area 18, is concerned with interpretation of visual sensation and storage of memories of visual symbols. Chapter 162, Occipital eye field, area 19, is concerned with reflex movement of eyeballs. It is also concerned with associated movements of eyeballs while following a moving object. Table 152.5. Applied physiology. Lesion in the upper or lower part of visual cortex results in hemianopia. Bilateral lesion leads to total blindness. Refer chapter 168 for details. Methods to study cortical connections and functions. By cutting or destruction of nerve cell. If the nerve cell body is destroyed, degenerative changes occur throughout the axon arising from it. By using Markey staining technique, course of the nerve fiber could be traced. If any part of motor area is destroyed, the degeneration of the fibers in the pyramidal tracts can be traced. If arm fibers are involved, the degeneration occurs up to lower cervical and upper thoracic level. If an axon is cut, the nerve cell body, from which the axon arises, undergoes chromatolysis. If any fiber in pyramidal tract is cut, the chromatolysis occurs in nerve cell body situated in motor cortex. Thus, this method is used to study connections and localization in motor cortex. It is also used for the study of connections of different parts of cerebral cortex. By recording electrical activity, evoked potential. When an impulse passes through a nerve, its route and the termination can be determined by recording the electrical potentials using microelectrodes at different points along the course of the nerve fiber. This method is used to trace certain pathways from or to the cortex, particularly auditory pathway and pyramidal tract. Evoked potential. Evoked potential is the electrical potential or electrical response in a neuron or group of neurons in the brain produced by an external stimulus. It is also called evoked cortical potential. When any receptor of skin or a sense organ, eye or ear, is stimulated, the impulses pass through the afferents and reach cerebral cortex. By using scalp electrodes, the potentials developed in cortical areas can be recorded. This method is used to determine the functions of various cortical areas. It is also used to map out the cortical representation of body, localization, for sensory function. Evoked potential is recorded by placing the exploring electrode on the surface of the head over the primary cortical area of the particular sensation. Indifferent electrode is placed on a distant area of head. In human beings, small disc-like electrodes are placed on different areas of head by using a tape or washable paste. Electrode cap, which is placed over the head, can also be used. Analysis and interpretation of the potential is done by computer. Evoked potential is characterized by two types of response. Primary evoked potential. When the stimulus is applied to the receptor or sense organ, the primary evoked potential appears after a latent period of 5 to 10 milliseconds. It includes a positive wave followed by a small negative wave. Primary evoked potential is highly localized and appears specifically on the cortical surface where the particular sensory pathway terminates. Diffuse secondary evoked potential. Finally, another larger and prolonged positive wave called secondary evoked potential is recorded. It is not localized. It appears on the diffused areas of cortex. Thus, the evoked potential includes P1N1 P2 sequence, i.e. first positive wave, first negative wave, second positive wave. Diagnostic uses of evoked potential. An evoked potential test determines the functional status of a nervous pathway. It also measures the time taken by the nerves to respond to stimulation. Intensity of response is also measured. Nerves from different areas of the body may be tested. However, three types of evoked potentials are commonly used in diagnosis. Visual evoked potential, which is recorded when the visual receptors are stimulated by looking at a test pattern. Auditory evoked potential, that is recorded when auditory receptors are stimulated by listening to a test sound. Somatosensory evoked potential, which is recorded when the somatic nerves of the limbs are stimulated by electrical stimulus. By physiological neuronography. When a small piece of blotting paper soaked in strychnine solution is placed over cerebral cortex, the nerve cells are stimulated by strychnine. The impulses discharged by these nerve cells pass through the axons and reach the termination in other part of cortex or other part of brain. By recording these impulses, the connections of cortex can be studied. By scanning. Nowadays, the functional activities of cerebral cortex or other parts of the brain are determined by scanning. Due to the fast development of technology, many sophisticated scanning methods are being introduced. Three such methods used widely are, computerized axial tomography, CAT, positron emission tomography, PET, magnetic resonance imaging, MRI. Refer Chapter 109 for details of these scanning methods.